So my name is Dr. Tom Johnson. I've been at UCL Biochemical Engineering since 2010. I started as an undergraduate student, and since then I've stayed here my whole career to the point now where I have my own research agenda, which is ultimately focused on producing better medicines. And something I thought would be very interesting for this talk was some of the research that I've been performing, integrating 3D imaging and 3D printing in order to do this. And with all research endeavors, particularly in the space of biochemical engineering, is often a very collaborative effort. So I work with a lot of people in academia and also in industry to make sure that this research is relevant and has some real impact on the real world. I'm part of a center of excellence or a research network with a company called Pall Corporation, who are a leading vendor in the bioprocessing and medicines manufacturing space. And they've helped us fund this center whereby we've got 10 plus PhD or EngD, which is engineering PhD projects, a couple of postdocs or PDRAs, and also myself in a fellowship role where I'm funded through UKRI. This allows me then to collaborate and work with all of these really exciting projects across a different range of biochemical engineering topics, and also interact with Pall Corporation, the company itself. I've also recently had some external funding through NXCT, which has allowed me to then set up these new exciting collaborations. And one of them is going to be with the University of Edinburgh, and that's going to be the 3D printing side that I'll speak about towards the end of this talk. So here's the contents of the presentation today. I'm going to start off by speaking about why we want to perform research in this biochemical engineering and also 3D printing space, before giving a very quick introduction to bioprocessing as a whole. In order to be able to 3D print these new exciting materials, what we need to do is understand how the existing structures and existing products on the market behave. And we've been doing this by applying high resolution 3D imaging techniques. And then using that information, we can start to inform how we're 3D printing these new materials. I'll then make some conclusions and also where we're excited to be going with this in the future. And on the right-hand side, you may be able to see this arrow or this length scale graph. And it's very important to note that all of this research and the discussion that I'm going to be talking about today goes across lots of different length scales. We're looking at whole capsules all the way down to what an individual nanopore looks like and everything in between, so that we can harmonize between all of these different levels, thereby getting the most out of our structures. Therefore, we can make the best medicines possible. And you've probably seen a lot in the news recently, and obviously highlighted by the vaccines during the pandemic, that there's a lot of exciting new medicinal and therapeutic products coming onto the market, and they present a lot of exciting clinical opportunities, life-changing and also life-saving medicines that are really changing the landscape of the previously incurable diseases we're now being able to start to treat. However, these new products are very complicated, and that complexity brings an inherent difficulty in manufacturing them, so that even if we can produce them in the lab, we need to be able to translate that into a scalable process so that we can actually produce it at the quantity that we need to treat a global population, and also efficiently at a price that we can actually afford. So there's no point in being able to produce a wonderful medicine if we can't actually then produce it at a cost that the NHS, for example, could actually afford to treat the population. And so my research focus has very much been on how can we better understand the structure of these materials we're using to produce and purify these exciting new medicines, and how can we relate that to the function and performance so that we can then improve them and adapt them for these new medicines. Now, just to give you a very quick introduction to bioprocessing, where in essence we're producing and then purifying these new medicines. And I have to say, this is a very simplified version of what we'd expect to see in reality. Normally, you'd see 10 plus steps rather than the 3 and 4 shown here, but this just gives you an overall idea of what we're going to be looking at today. So on the left-hand side, we have a bioreactor, which essentially is where you're going to be producing your new exciting medicines. And typically that's done by growing cells, and then either the cells of the product themselves, the cells produce a product and then they secrete it so that the product's in the liquid or the broth, or the cells produce the medicine inside of the cell, and then you have to break it open. And all of these different modalities cause several problems. When we're talking about biological material, we're not looking at nice, clean liquid. We're looking at things that are full of these cells, full of these different proteins and all of these other things in the cell broth. But we have to then purify out, because we can't just put that straight into a human for a medicine. We need to make sure we can purify it to a really high quality at a high yield. So what I'm going to do today is focus on 3 steps, giving some basic understanding of what's going on in the purification train, and then how we can then start to 3D print materials based off of that information that we've gathered. The first step would be depth clarification or depth filtration. And what we're doing in this step is removing the large cells from our broth. That then allows us to just have the smaller protein or products in our liquid feed. Having removed all of the cells then allows us to perform purification to a much higher standard, particularly during chromatography. Now, you may remember from school, you may have done paper chromatography, separating out various dyes from an ink is a good example. And then the chromatography that we use for bioprocessing is a much more advanced version, but it's still ultimately trying to do the same thing in terms of separating out things that are of similar size from each other. And so what we can do is use various different ways of looking at this, for example, through charge. So we can separate out things that are the same size but have slightly different chemical properties, and then that will allow us to separate out those impurities from our intended product. And the way that we do this is that we have a cylindrical column that has a series of microporous spheres, about 50 micrometers or a 20th of a millimeter in size. And these spheres will then bind our product, and then our impurities and the things we don't want will flow through. We can then change the pH or the conductivity, which then pushes off our product, and we can recollect that now concentrated product in a pulse. You can also do it the other way around, where you let the product flow through and you capture all the impurities. But only what you're trying to do in this stage is make sure you're just isolating your product at a very high quality and at a very high purity at once. We can then move on to the final stage of sterile filtration, which essentially is making sure that the product that you've produced and purified is free from bacteria or any contaminants like that before you put it in a vial at the end, or whatever the product looks like. So what we're going to do is going to go through 3 short case studies looking at these different materials, looking at how their structure relates to function and performance, and how we can then use that information to inform the 3D printing of these new structures. And the way we look at the structure of these materials is by applying high resolution 3D imaging. And there's 3 main techniques that I've used in my research. The first being X-ray computed tomography, and I'm sure you've all seen a 2D scan or a radiograph from X-ray imaging if you've ever had a broken bone. Through computed tomography, what we're able to do is not just take 2D images of what the material looks like, but actually contextualize that in a 3D volume, allowing us to see inside these materials at a very high resolution without actually having to cut the sample open and therefore change it. On the bottom, we've got a couple of examples. So we've got that chromatography column on the very bottom left, and we can see through that. And as I said, there's also these microspheres that make up that cylindrical column. And what I have to do, and I was actually doing some of this in the lab yesterday, is isolate an individual microsphere and put that on top of a pinhead to perform nanoscale resolution imaging. So that gives us a huge amount of information. And UCL was one of the very first places to be able to have this technology and this X-ray CT equipment at the nanoscale. However, it is very tricky and very fiddly, as you can imagine, trying to put one of these small things on top of a pinhead for imaging. Now, another technique we have is focused ion beam, which is very similar to scanning electron microscopy. It again gives you a very high resolution image. However, SEM typically is restricted to the surface of a sample. So what we can do by applying focused ion beam is cut through these materials and then get another 3D picture. So we've got 2 techniques which we can compare to look at what does the structure look like inside of our bioprocessing materials. Whilst these are both great for looking at the high resolution detail and all of the individual pores, they're not particularly fantastic in actually looking at what does the material look like when it's used. How does all of that product or impurities, where do they actually get stuck within these samples? And so the third technique is confocal microscopy, which is perfect for doing this. So by complementing these 3 techniques together, what we can do is understand what's going on within our structures and where our material is getting stuck as well. Moving on to depth filtration or depth clarification, the early stage of the downstream bioprocess. On the left-hand side, we've got an example of a filter capsule. We then zoom in to an individual layer of this depth filter material, 5 millimeters or so thick. And then we can take it down to another level. Again, we're starting to see that individual porous structure inside of this material. And a paper that I produced a couple of years ago in 2021 spent a lot of time optimizing the image quality, because we need to make sure that what we're seeing truly represents what the actual material looks like. And we performed a lot of analysis. How does the structure change as you move through the material? and also the image with the yellow lines at the bottom. How does the flow behavior of our products and impurities actually happen within these materials? So how can we get the best separation for what we're trying to achieve at this stage? Then Maria Peral led a piece of research that we also published recently, looking at how do these depth filters actually change as they start to get clogged up. So as I said, these are at the very start, they're dealing with very rich feeds, lots of cells. So you can see in the top image on the right-hand side, these nice clean materials, and then on the bottom, how they start to get used and how they degrade as they start to be used across their life cycle. This then allows us to better, again, demonstrate how their structure relates to function and performance, informing our 3D design cycle that I get onto in a moment. But something that's really interesting with these depth filters is it's not just pores or space and then material, but actually there's multiple materials within our material phase. And so using X-ray CT, we're actually able to identify all of these individual materials, which I think is really cool within these structures. So on the left-hand side, we've got this black voidage phase, this dark gray, which is a cellulose, which is the base matrix, which comprises most of the structure. And then we've also got these really bright white areas, which is perlite in this case. That allows not only to support more of this structure because it's very soft being cellulose, but it also has some binding action as well. And using these 3D data sets, we're then able to color code these. So we've got the black, the blue for the cellulose, and then the bright orange for the perlite. And again, perform lots of detailed analysis, which all informs our design cycle for 3D materials in terms of how does this depth filter behave? Can we use any of this information in the future to design our new structures? Moving on to chromatography, again, the main purification step within downstream bioprocessing. As I said, it consists of a cylinder, which we've got on the left-hand side, digitally cut through that to give you a horizontal slice where you can see all of these individual small spheres. Now, a lot of work we do in chromatography is based on a lot of assumptions in terms of how these materials behave. Such assumptions include that all of these particles are perfectly spherical, they're all of exactly the same size, and they all sit nicely in rows and columns on top of each other and next to each other. However, by using X-ray CT, by looking at a real available product that we've got in the lab at UCL, we can start to challenge some of these assumptions, actually see what's really going on within our bioprocessing structures. Not only that, in terms of producing pretty images, we can also then start to perform 3D analysis and positional-based evaluation of these structures. So we've got a graph on the right-hand side. I'm not going to go into too much detail, but suffice to say, it's been known for decades that there's a difference in the porosity or the amount of 3D space between all of those microparticles between the edge of the column, represented in white here, and to the center. And by having 3D imaging approach, we're able to then identify this difference between the edge and the center for a range of different materials. We're then very interested to look at what these actual microparticles look like when we start to image them on top of the pinhead, because they've got an internal porosity, and that gives you a very high surface area available for binding your product to your impurity, which is really important to get a very high capacity. Again, we can start to look at lots of different materials, ceramic being a good example. You can see in this case, it's not particularly spherical. And we can also look at cellulose again, which is a very common material alongside agarose due to its high biocompatibility in this space. You can see in the central image that we've got these large spherical pores within this structure. However, at a resolution of about 50 nanometers, we weren't able to see all of the detailed structure. By improving that image to a high resolution scan, we could then identify all of these individual or larger pores and also those fine features between them as well. So we're getting a huge amount of detail out of these structures, understanding how they can properly bind them, what are the diffusion mechanics within these materials to best understand how can we separate our structures and our products of the future. Won't go into the graph, but again, suffice to say, we need to make sure that we're getting a sufficient resolution to be able to see everything we want to image. And then, but we don't want to go too much further, because if you keep pushing it further, you start to lose lots of different other benefits that you get from 3D imaging. So it's very much a balancing out. And to understand in a very similar way to the depth filters, how do these materials change as they're used during their lifetime? On the left-hand side, we started to compress these materials. Now, some of these chromatography resins can cost thousands of pounds for a litre. And as you start to get to industrial scale, what you don't want to do is start to damage them or compress them. As you compress them, you're ultimately changing the structure between the beads, And if you irreversibly compress it, as we can see in the central image, you could actually end up spoiling and throwing away not only the product you're trying to purify, but the actual materials and the chromatography resins as well. So designing something that's not only biocompatible, but also compression resistant is very important. And again, the bottom left-hand side graph, I'm not going to go through it in too much detail, but we're able to not only visualize, but also characterize what was going on in terms of this compression within these cellulose chromatography columns. And then as part of a collaboration with IIT Delhi, we are able to also then image these materials using SEM. So you're on the right-hand side, you've got a couple of images. The left-hand side one is of a clean chromatography resin with all of those micro and nanopores. And then on the right-hand side, after it's been used for a certain number of cycles. And because we're dealing with biological materials, what happens is you start to get lots of fouling or clogging of these larger materials and particles, which start then inhibiting your product or your impurities from getting inside these chromatography materials. And in this study, we were able to then correlate what was going on in terms of the images we were getting to the performance of these materials. Again, very important to be able to properly understand that so that we can then design new 3D printed materials that can combat some of these issues. The final step is sterile filtration. And for products such as proteins and monoclonal antibodies, which are really the golden standard of bioprocessing medicines for a lot of decades, this is a reasonably trivial step. You're just ensuring that there's no bacteria that goes through these materials, goes into your final vial or your final product form. However, what happens when we start to look at these new products? They're far more fragile, they're far larger, and their size actually starts to get much closer to the pore size of these sterilizing filter membranes. So previously, when we're starting to look at these steps, it's very trivial before, but now we really need to start to think about every single step because these products are very valuable and very expensive. So we don't want to be throwing it away at the end of our process, given we spent so much time and resource on actually producing and purifying it in the first place. Very similar to the individual chromatography spheres, on the left-hand side, we've got a very small piece of this sterile filter membrane, which is on top of a pinhead. You've got these blue and red boxes, which is basically where I'm scanning using X-ray CT to look within this material. We looked at two different types of membrane, both of the same material of construction, PES, but they've got two very different characteristics. On the left-hand side, you've got a very asymmetric material with a much larger pore size. And what we could see then is that the structure actually changes as a function of distance through the material. So at the top with these pore network maps, you've got these red and these orange pores, and then it starts to get tighter down to these blue pores at the bottom. And using this approach, using 3D imaging, we can identify all of the individual pores, their size distribution, which we see on the right-hand side, and how they all connect. And we can start to relate that to what's actually happening within these materials. And we can compare it to the lower membrane on the right-hand side, which has a far more consistent structure. And again, we can see that represented in the pore network map and also the size distributions. As I said at the start of this talk, something that's very important is not only understanding what the structure looks like of these materials, but where our product and where our impurities are also getting stuck within them as well. So confocal microscopy is the technique we use, as I said earlier. We've got this green dye, which we're encapsulating inside of these liposomes, which are one of these new exciting medicinal delivery vehicles. What this allowed us to do when performing confocal microscopy is start to see where these liposomes were getting stuck within our within our membranes. And then we could then relate that position to what does the pore size look like, etc., etc. Something that's also very interesting for these materials is how can we relate to that to the conventional processing function. So we know what the yields are and how these behave as we start to change the pressure that we're using to drive through these materials. How does the change in pressure improve the yield? We were finding that at a lower pressure, we were getting a 90% yield, and at a higher pressure, we were getting 99, 100% yield, which is fantastic. So what we can start to see is how does that yield also relate to where these liposomes that are getting retained are getting stuck within these structures. And so we found that whilst you could improve the pressure, gave you better performance, and we could see where the remaining liposomes were getting stuck, this step is ultimately ensuring that you've got no bacteria in your final step before you go towards your vial or your final filling. And what we wanted to see is that if we put a lot of bacteria in these samples, what happens in our real bioprocess? And on the right-hand side, we can see that there's actually some co-filtration mechanics that were going on with these new products. So even for these reasonably trivial steps, we've got to put a lot of thought into our design of our materials to ensure that what we're designing gets the best separation possible across all of these different steps. This leads me on to the 3D printing side. So now we can understand how all of these purification materials relate to function and performance. We can understand what's good about them and also some of their existing limitations, given that they're using technology that's been around for decades. 3D printing and combining that with 3D imaging is extremely exciting because now across all of these different length scales, what we can do is perfectly tailor the chemical and physical attributes of what we want to do in our separation stage to the product and also the process stream of interest. So we can see what's the size of the product, what's the right pore size to then best separate that product from the impurities in that specific process stream. 3D printing and 3D design gives you a huge amount of opportunities in this space. So I'm going to now present some work that it was really hot off the press, and we've just recently submitted this for publication as part of a collaboration with the University of Edinburgh and Dr. Simone Di Martino, who's a real world leader in the 3D printing space for biopurification. And we can design perfect structures, as I said, but what we wanted to do is combine that with 3D imaging. So what we're designing is perfect, but are we actually fabricating that using 3D printing? And in this case, we could design these materials and they've got the same material phase and the same pore phase in each case. All we're doing is just going from larger material and pore phase to a tighter material and pore phase as we go from left to right. So the structure is concerned, all we're doing is just changing the size of each of the individual units. And we can see as we start to push on the top hand side, we've got the designs and they're all perfectly designed. But as we start to print these materials and as we start to fabricate them at very high resolutions, close to the printing capabilities, we start to see some artifacts and some problems. So what you don't want to do is design the perfect material and then not be able to actually 3D print it in the first place. So what we're able to do using 3D imaging is to develop a design cycle, therefore looping back in what we're actually seeing from our real structures in the lab back to the design stage so that we can then improve it there and we can go through a cycle again and again. Got a couple of short videos which seem to be working, which is great, whereby we can then see this 3D detailed structure of the coarser material in this case, and also on the right hand side, one of the finer structures. So you can see both that material and that pore phase. Now, something that I thought was very interesting and what we didn't actually expect in the first case is you may be able to see some horizontal lines going through this material, which are very bright. Those are actually all of the individual 3D printed layers that are formed as we 3D print the structure, which I think is very exciting. Now, if you remember, we had the chromatography column, which consisted of all of these individual microspheres. And if we have a comparison of the whole packed bed on the previous slide, we can now start to look at individual units that we're designing within these structures. And as I said earlier, something that's very important is that we need to ensure that what we're designing is exactly what we're printing. And so if we look at an individual subunit, we can design it, we can then print it, and then we can then image it and then see and then directly compare it. So the blue image is the CAD file that we've produced. We then 3D print that. On the left-hand side, you've got the raw material. The second image is of the binarized material to pore and porous phase. And then on the very right-hand side, we've got the overlap. So you can still see there's some discrepancies between what we're designing and exactly what we're printing. And if we're really trying to perfect the chemical and physical attributes, we need to really make sure that we're getting those harmonized and aligned. And what we've been able to do using this approach is really bring those tolerances down so that the blue CAD file gets very close to what we're actually 3D printing in the first place. Now what we can then do, now that we're looking at these materials, is actually then see how does the flow behavior occur within these structures. We need to make sure that we're getting very even flow distribution through these 3D printed materials, because what you don't want to do is have this expensive product and then be purifying it in an uneven fashion. And across these three different sizes of the units that we looked at, we got very good flow agreement on the real samples that exist in the lab, demonstrating to us that the structures that we were producing were actually performing as we expected. Now, moving down to that final length scale, as I said, for inside of those chromatography microspheres, you've got this nanoporous structure, which gives you a really high surface area available for binding. So all of these 3D printed materials, through some very clever chemistry, are able to actually pull out an internal material phase Thereby, we've got nanopores within our structures as well. Very similar to those chromatography beads as well. We've got a smaller amount of material put on top of a pinhead for nano CT scanning. The second image is the interface between the pinhead and then the material on the left-hand side. And then we can image that at a very high nanoscale resolution. Both represent that in a 2D version on the third image and a 3D volume on the right-hand side. So now we've got an understanding of what's going on in our 3D materials, all the way from the whole structure down to an individual nanopore and everything in between. We can then perform analysis in a very similar fashion to what we saw from our previous case studies. So we can look at the diffusive flow behavior, both for the bulk on the left-hand side and also for individual particles, which are represented by these red streamlines going through the material in the second image. This is really, really important because the products we're starting to purify now in these new exciting medicines are far larger, so we need to start thinking about how they behave as particles within our materials. And in a very similar way to how we saw on the sterile filtration case study, we can start to identify what's the size of all of these individual pores, how do they all connect together, and what's the pore size distribution within these materials. So we can start to say, okay, so what's the right structure that we're printing, and what's the right chemistry that we're pulling out of these materials to make these nanopores to get the perfect chemical and physical structure for the product and process that we're interested in at the time. And we're hoping in the future this will allow us to then greatly improve the yield and the product quality and the product purity of these exciting new medicines going forward into the future, so that we can really translate from the lab to a scalable manufacturing process, so that ultimately that benefits patients that are going to receive these really exciting, life-changing, potentially life-saving medicines going forward. So to make some conclusions, 3D printing is an extremely exciting technology. It's certainly got some way to go so that we can fully apply it, but it gives us a huge amount of opportunity to take our understanding of what's going on within our bioprocess and then be able to specifically tailor those materials to our product and our process. And the 3D imaging is very powerful because it gives us so much understanding of what does a real material look like, what does a real chromatography column look like, what does the 3D material actually look like when you start to look at the nanopores. So we can relate it back to functional performance, thereby we can continually improve the quality of the 3D printed structures that we're actually producing. And we can continue to apply this to real medicines, and by collaborating between academia and industry, we can make sure that what we're looking at is really clinically relevant and impactful. Some references and some of the research that we've published previously, both from UCL and as part of collaborations with external partners. And finally, just to say, as I said at the very start of this talk, a lot of research that's performed at UCL is not performed alone, and there's a lot of collaboration that goes on. So I'd like to thank my friends and colleagues at the Department of Biochemical Engineering and Chemical Engineering at UCL, my industrial sponsors at Paul Corporation, who have performed a lot of the funding and also expertise in this space, and also to my collaborators at the University of Edinburgh and the DiMartino Lab, who have been fantastic, leading 3D printing and really exciting for what we're going to be doing in the future. My email is available on the top right-hand corner. So finally, thank you very much for listening. And now, Kim, I believe I'll hand back over to you.